This particular field represents a typical bronchiole cut in cross section. Its characteristic features is the scalloped or folded nature of the lining epithelium. This is perhaps due to the contraction of its major component of a supporting wall, that is this bronchial smooth muscle that's seen cut a little bit lengthwise in cross section and usually courses all the way around the outer circumference and forms the supporting wall to this small tube. The other feature to note is what is absent. The hyaline cartilage support has disappeared, the glands have disappeared, and there is very little, if any, lymphoid organ or tissue in mass present at this particular point. Note also carefully at the epithelium at this particular juncture. It is now definitely a very characteristic simple columnar epithelium, which may or may not maintain cilia at this point. Some of these tubules will. That same bronchiole, as examined at low power on the previous section, showing in detail its lining epithelium. A simple columnar form that's getting fairly low. Not many cilia are observed, and no goblet cells are visible at this particular point in its subdivision, as we're nearing the terminal end of this conducting portion of the respiratory tree. There's a clip through the supporting wall of the bronchiole, that is that bronchial smooth muscle. A few of the cells, though it's difficult to demonstrate, do retain their cilia, but no goblet cells are observed at this particular point in this section. Here's a couple more sections of smooth muscle as it has a spiraling course around the bronchial tree. And one can see it's a simple columnar epithelium getting lower in stature with very few, if any, goblet cells present. So we're down in the near the end of a typical bronchiole. The goblet cells will disappear and another cell will make its appearance, a Clara cell. An additional section through a bronchiole that's cut perhaps a little more crisply and more of a perfect cross section. It illustrates quite a bit better the bronchial smooth muscle, how it forms a complete spiral around and forms the supporting wall of this particular portion of the conducting system of the airway, as well as the folded nature of the epithelium, which even at this magnification, using the intermediate power, one can see as a simple columnar form. Goblet cells at this point probably aren't present. There should be a few ciliated cells, and at this point a specialized cell known as the Clara cell should begin to make its appearance. This particular field is a scanning electron micrograph showing the mucosal surface as viewed from the surface of a small bronchiole. Here's one of the bronchiole folds shown here, and it's crossed by the arrow. The arrow crosses another one of these bronchial folds, and another is shown here. So this is just a small portion of the bronchiole wall, looking at the tops of the folds that were seen in previous sections. What this scanning micrograph actually shows you are the ciliated cells. The tops of them are just their cilia. You're not seeing the apices of these cells shown here. Some here, some here, some here, and scattered around. In addition, it is showing the tops of other epithelial cells forming the epithelial lining of the bronchiole epithelium, and these perhaps could be Clara cells. Nonetheless, at this point, all one can say these are the apices of several non-ciliated cells together with the ciliated cells in this small bronchiole that's being observed. This structure here, though it's folded over, it was an erythrocyte, and it gives one a little bit of perspective as to the size of these cell apices. A small portion of the bronchiole epithelial lining as viewed from the surface in another scanning electron micrograph, but one that was taken at increased magnification, simply to illustrate in greater detail the dome-like apices of the non-ciliated cells, as can be demonstrated here, as well as demonstrating without much question some of the cells possess or retain, even at this point, this far down, numerous cilia. And this is what the individual cilia look like when seen from the surface, and you can see there are groups of them indicating they're all coming from a single cell, like this cluster here, coming from the top of a specific cell. So this is the surface lining epithelium of a bronchiole, but just seen at increased magnification. The previous bronchiole as seen at high magnification, illustrating the folded nature of the mucosal surface to the luminal wall and the supporting wall of smooth muscle, which extends from here to here. And one can visualize there's just a slight amount of laminar propria supporting the overlying epithelium. Note that the epithelium is simple columnar in nature. A few scattered ciliated cells are present. No goblet cells are present in this particular case, so it's far enough down so the goblet cells have ceased to make their appearance. And as mentioned previously, the goblet cells will now be replaced more or less, or their position taken by a specialized secreting cell known as the Clara cell. So this is a typical portion of a bronchiole and its characteristic features. The supporting wall only of smooth muscle and this folded nature to the epithelium. The surrounding structures as seen now in the field of view are collapsed alveolar walls. So these are the alveoli, these spaces out here, they're alveolar walls as indicated by the pointer. To give you an appreciation for the differences in size. Another alveolus showing portion of its lumen shown here, another is shown here, another here as well. So this is a portion of the wall of a typical bronchiole as seen at high magnification. Finally, one will get to the end point of the purely conducting portion of the respiratory tree at a terminal bronchiole. One of those is shown in the field cut more or less in cross section, but it's somewhat at an angle. One can see that the lining epithelium is more cuboidal in nature. We'll see that most of the cilia have disappeared. There should be no goblet cells present, but yet wall is supported by a layer of smooth muscle, this bronchial smooth muscle wall, giving it its support. You can see as the arrow indicates, as it spirals around the lumen, it then starts to form a more open spiral. We're missing a bit here. Here the smooth muscle musculature is cut in cross section, and then continues in this direction. This terminal bronchiole rapidly gives rise to the first portion of the conducting system. It's a transitionary portion, a very short segment, known as the respiratory bronchiole. And so this tube, then angled perhaps either above or below the field, entered here and then is going in the direction of the arrow. That is the position of the lumen of this particular tubule. And let's trace it. It has a fairly decent lengthwise cut through it, down to about where the arrow now indicates. What one will see is that the bronchial smooth muscle will now open up and form a more open spiral, a corkscrew type of arrangement. And if one just takes this area here, this is a cross-section of that smooth muscle bundle as it forms this open spiral. There's another one here. This is a segment from an adjacent structure. There's another bundle of muscle here. And if we look on the upper surface, a bundle here, a little bit tangentially cut here. Here's that spiral cut several times. The one on the lower surface is cut here as it forms this open spiral. One forms here. And so you can see how this bronchial musculature is spiraling out and beginning to fade and disappear, and will finally end in some of the more in the alveolar duct area. Now, as this is occurring, as that corkscrew arrangement rather than the tight coil of smooth muscle as was seen as it's observed in the typical bronchiole, in these gaps between the smooth muscle bands will be the initial appearance of alveoli lined by typical type 1 and type 2 pneumocytes. In this transitionary portion, that is the respiratory bronchiole, usually the epithelial cells covering the smooth muscle wraps will retain sort of a cuboidal type nature and mimic those cells, or the epithelium will mimic those cells that have lined the terminal bronchiole. So these should show columnar or cuboidal type cells, really. As you can see, there are cuboidal type cells here. Here you can see it's very, very thin and thinned out, so these are probably type 1 alveolocytes, a very thin, flattened, squamous type of epithelial cell. And this is that short transitionary period that will eventually go and become, that is, the lumen will become continuous with an alveolar duct, which we'll examine shortly. Let's go back and examine some of these points at increased magnification so the details can be observed. The terminal bronchiole, as seen at increased magnification, shows the simple cuboidal to columnar lining type of epithelium. The retaining smooth muscle wall, though it's beginning to form a spiralar type structure. And these vacuolated, non-ciliated cells at this point may indeed be Clara cells. So sort of a bubbly, vacuolated appearance, non-ciliated, though there could occasionally be cilia this far distally. There's definitely no goblet cells and definitely no bronchial glands, which disappeared much, much more proximally. Now, if one follows this section in the direction of the alveolar duct, one can see the epithelial lining on this particular surface. These cells are quite likely Clara cells, the sort of bubbly type of appearance. Here, once again, we can see at increased magnification this spiraling, looping, this open position of the bronchial smooth muscle as it courses down. And one can make out over this particular cross-sectional profile of the spiraling muscle, in between the muscle masses, one can see the epithelial lining changes from this cuboidal to this little area right here. This little fine line is the combination of an endothelial cell and a type 1 pneumocyte. This is the thinness we're talking about. So this is probably, though it's not well represented here, an alveolus here. The cuboidal lining epithelium of this respiratory bronchiole shown here, and though it has exudate and some material in it, another alveolus is shown here. And this type of a transition will repeat itself over and over again. Here we have the alveoli that are stretched out. You can see they're this is the luminal lining, this is the capillary. This surface here will be covered by type 1 cells as further down. And then we get perhaps another little bunch, though it's ragged, of these cuboidal type cells that were jagged up. And that's about the length. They're a very short transitionary period going from the conducting to the respiratory portion of where gaseous exchange takes place of the lung. So this is just the very terminal part, the end of or a transition between the end of the conducting portion of the respiratory tree and the purely respiratory portion where gaseous exchange is actually going to take place. And so what we're visualizing here is a portion of that transition, that is the respiratory bronchiole. And back up finally to a terminal bronchiole as shown here. An additional segment through another respiratory bronchiole, showing you the spiraling nature of the bronchial smooth muscle as it opens up. This is the typical cuboidal lining covering epithelium, and then if we trace it down slightly, you can see how they're ballooning out. The epithelium becomes much, much thinner in these little outpouchings and the initial appearance of the alveoli. Then the bronchial muscle appears again here, it jumps here, it jumps over here, and usually, but not always, on these smooth muscle bands as they project sort of into the lumen of these respiratory bronchioles, they're going to be covered usually by a cuboidal type of epithelium. In this particular case, this does not appear to be the case. It looks like an alveolar type 1 cell that's sort of stretched out and entering the lumen. So these short transitionary points, this is what is being presented as a respiratory bronchiole, this very short transition point between the conducting and the appearance of alveoli, and then a purely respiratory region, as one will encounter in the alveolar ducts. This particular field represents a region of lung taken from a mouse. The mouse was his lungs were inflated prior to its being terminated and fixed in an inflated condition to demonstrate the alveolar ducts and alveoli, which would be impossible if one excises just the lungs, and due to the reduction in pressure, the alveoli simply collapse. What this particular tissue shows, the remainder of the lung is atypical, but from the alveolar duct to the alveolar sacs, it's fairly typical of all mammals. What this illustrates, this particular tissue, as an alveolar duct as it runs along the course as indicated by the arrow, and then divides at this particular point. The alveolar ducts join with the respiratory bronchioles and then will go proximally. Distally, they will terminate in large sac-like structures known as alveolar sacs. So these tubular type structures, or so they appear, are the lumina of alveolar ducts. The alveolar duct is simply a tube, its wall of which is made up of successive alveoli, as indicated by the arrow. Here the alveoli have been cut more or less through their center and shows their opening in continuity with the lumina of the alveolar duct, which is shown here. So this would be an alveolar duct. This is an alveolar duct, or the lumen thereof, and its wall made up of successive alveoli. Another is shown here. These are usually tubular-like structures, but because of the contorted nature and the branching of the alveolar ducts and the rest of the respiratory tree, you see them cut at all total different variety of angles. So this would be an alveolar duct coursing this way. There's another branching and going this way. Another one is flowing in that direction. So with this scanning objective, one can see numerous of these long tubular structures, the wall of which is made up of successive alveoli. These are the alveolar ducts. One shown here, another here, another here. This is probably one that's overinflated and would be cut in cross section. So this is what is being indicated or talked about. These long tubular structures, or the lumen looks like a long tube, but its wall is made up of successive alveoli, the alveolar ducts. These eventually terminate in a rounded, more sacular type of structure known as an alveolar sac. It sort of like ends like a little cluster of grapes if you cut it in that fashion. And this is where the respiratory passage will terminate in a succession of alveoli. This is perhaps one here, but cut at an angle. If one can envision this alveolar duct coursing like so and coming here, then it'll end in maybe 5 or 6 alveoli, and this is where the epithelial lining will actually stop. And that'll be the end of this branching tubular system. This is perhaps a better demonstration here, showing an alveolar duct coming down and then terminating in this sacular-like structure. So these are the alveolar ducts as demonstrated in an inflated lung of this particular mouse. This particular tissue is only good, that is the mouse tissue, for looking at alveolar ducts and inflated alveoli. Once that has been accomplished, examine the other typical lung section. Increased magnification of the alveolar duct in the mouse lung to demonstrate the alveolar duct, showing you indeed that it is made up of successive alveoli. And note the very thin nature of the wall of the alveoli. Here we see one that it's cut in cross section. Actually, what makes their walls easy to visualize is not so much the lining epithelium within their walls, but the erythrocytes within the capillaries. These small ghost-like structures forming the background and giving some sort of contrast so these alveolar walls can be visualized are erythrocytes within the capillaries within inside of the alveolar wall, which is extraordinarily thin. And one can appreciate that by looking at these at a slight increase in magnification, then actually visualizing the erythrocytes coursing within the capillaries forming within the alveolar walls. one gets a very good indication of the very delicate nature and of these particular structures, that is, the alveoli. Here is a group of alveoli as seen in cross-section. One is shown here, another here. All this lacy type of tissue with the lumen within its inside, its surrounding, are alveoli, but alveoli that have been cut at different angles. Again, another field showing you the mass of erythrocytes, and if one traces those, that is what's really outlining and making the alveolar wall visible in these many of these tissues, particularly the individual alveoli. This is a portion of lung in the plastic embedded material and stained with hematoxylin eosin and viewed at high magnification. What is shown in the field of view is the alveolar wall, which is coursing roughly in this position. Another alveolus is shown here, another one up here, and so this is that lacy type of material one sees in the background. But looking at this expanded alveolus, several features can be observed. First of all, this is the alveolar wall here. What's interesting and what can be visualized at this high magnification with the plastic type of material is this thin membrane coursing here and illustrated at the tip of the pointer. This is the blood-air barrier, and it's going to consist of an overlying type 1 epithelial pneumocyte, or the cytoplasm thereof. In between the two will be a basement membrane, and also forming this little thin hair-like membrane, of course, will be the endothelial cytoplasm. The nucleus of the endothelial cell is shown here, as indicated by the pointer. So this illustrates the thinness of that blood-air barrier where this exchange of gases is occurring. It can either be 3 or 5 usually. An epithelial barrier on the lining the lumen of the alveolus, that is the type 1 pneumocyte. Its basement membrane, perhaps the basement membrane of the endothelial cell, and then the endothelial cell cytoplasm itself. Usually there's a fusion of those two membranes. If not, that is the fusion of the two basement membranes. If not, there's a little connective tissue interval between the two, and you will end up with a 5-layered system, but it's all extraordinarily thin. Now, if we look up into the wall, this capillary happened to be empty. If we look up further along, you can see erythrocytes within the alveolar wall, but one can still make out under very careful observation that little thin hairline membrane just holding in the erythrocytes. So again, this is that blood-air barrier, the combination of the type 1 pneumocyte and endothelial cell cytoplasm. And over on this side, one can see this capillary is empty. There's an alveolus over here. This capillary adjacent to it is empty as well. So this is the dimensions that are being talked about, an extraordinarily thin barrier where these gaseous exchanges can occur at such an extraordinarily rapid rate. Here again, though, it's not very well represented. This is sort of a collapsed alveolus here. Here again are 2 erythrocytes, and then that very thin barrier shown here. This rounded cell, though it isn't well represented here, is a type 2 pneumocyte. They usually have a frothy appearance. They're sometimes countersunk down within the alveolar wall. There's another collapsed alveolus shown here. This is another example here, perhaps better, of a type 2 pneumocyte. The type 2 pneumocyte located here generally has a cuboidal type shape. It has this, its cytoplasm is filled and appears vacuolated with these like little myelin bodies. It has this vacuolated appearance. These myelin bodies or whorls of material are thought to represent surfactant. So this is one shown here, a type 2 pneumocyte. Another one here that is sticking out, actually protruding into the lumen of the alveolus. If one goes a little bit over on this side, here again is that little thin blood-air barrier made up of the endothelial and type 1 pneumocyte with an intervening basement membranes and connective tissue. So on the plastic type of material, these critical cell types, the type 1 and type 2 pneumocyte, can indeed be observed with careful observation. An additional region of lung shown at increased magnification, showing several collapsed alveoli and illustrating some of the features of the epithelial lining. Once again, this is the lumen of a collapsed alveolus shown here, and once again, just to drive home the point of the thinness of this epithelial lining and its association with the endothelium. So this is that very thin strand right at the tip of the pointer and the underlying erythrocyte. A little gap in between, that's light. So this little dark interface is that blood-air barrier, consisting once again of a type 1 pneumocyte, the cytoplasm thereof, basement membranes perhaps fused in between, and then the endothelial cell cytoplasm lining the alveolar capillary, which is here. So this gives you again another good indication of the extraordinary nature of this barrier. This is an empty capillary shown here where it bulges out somewhat, and you can see the erythrocytes that were coursing through and were coming in this direction. And here's another example of that barrier showing here, this very thin line. It's almost like an interface. These cells, as shown here, and countersunk in, that has a vacuolated appearance, they're more round or cube-like in nature, are type 2 pneumocytes, and these are the cells that are producing alveolar surfactant and trying to reduce the surface tension within the little fluid lining the interior of the lung. So this is an example of a type 2 pneumocyte here. Another is shown here. Another is shown here. All characterized by their more or less round or cube-like appearance, so oftentimes countersunk into the alveolar wall, such as shown here, and always show this vacuolated appearance with these myelin bodies, these so-called surfactant-containing bodies within their cytoplasm that can be demonstrated quite readily using the electron microscope. So this is a series of alveolar walls that have, due to the lack of pressure when this tissue was excised, was cut out and has this rather confusing appearance. But several of the detailed features can be made out when you examine this particular tissue at increased magnification. Again, that blood-air barrier demonstrated quite well at the tip of the arrow. This particular image illustrates a three-dimensional view of the parenchyma of the lung as seen with a scanning electron microscope. So this whole region is a region of lung that was excised from a lung and then now viewed with the scanning electron microscope, so you can see surface features. What is being illustrated are numerous alveoli with the alveolar walls. Here we can see the base of an alveolus with this lacy interconnecting network are all alveoli that are closely associated one with the other and one sort of joining with the other, giving you this sponge work of tissue. The spongy nature is so characteristic of lung tissue. A small bronchiole is shown here to give you some idea of the scale of the size. So this is just a three-dimensional network which hopefully one could visualize in their mind's eye when looking at some of the sectioned material of numerous alveoli. Each one of these little empty spaces as an alveolus, as seen in this three-dimensional imaging using the scanning electron microscope. So this is simply lung parenchymal tissue seen at a relatively low magnification. This particular figure illustrates a scanning electron micrograph examining the interior or lining surface of an alveolus. What it shows are the following features. This line extending across the field and being traced by the arrow, it represents the boundaries, the fusion points, or the junctions between type 1 pneumocytes. So this flat, smooth area now being crossed by the arrow is the cytoplasmic or the plasma lemma of a type 1 alveolocyte. Another junction is showing here where it joins another type 1 immediately adjacent to it. Another boundary is shown here, that little raised line. So those are the junctional complexes that bind type 1 cells together as seen along the surface. Another is shown coursing this way. These round appearing cells that have that fine microvillus appearance to the top, these are the apices of type 2 alveolocytes, those cells that secrete surfactant. Another one is shown here and is being crossed by the arrow. Another one is shown up here, and two at this location lying immediately adjacent to one another. So the type 2 cell usually, but not always, is countersunk somewhat within the epithelium and are these cube-like cells that one sees in section. Here we're just seeing the apex, the very top, and showing the its junction here, this light raised line with adjacent type 1 cells that are showing here. So this is a rather unusual view, taking the scanning electron microscope and actually entering into and looking at the surface features of the interior surface of an alveolus. This particular scanning electron micrograph also illustrates the lining interior of a alveolus. What's interesting about this particular figure is that the junctions between type 1 cells can be seen at the tip of the arrow. So all of this region in this particular area, despite its contorted appearance, and then down along this surface, is all covered or lined by a type 1 pneumocyte. These two structures, as was illustrated on the previous electron micrograph, are the apices of type 2 pneumocytes. What's interesting in this particular electron micrograph is this structure here. This is the outline of an erythrocyte, or a red blood cell, coursing in the capillaries beneath the alveolar wall, or within the alveolar wall, beneath the type 1 epithelium and beneath, obviously, an endothelial cell. Yet this wall is so thin you can see the outline of the erythrocyte as indicated by the arrow coursing along within the alveolar wall, but yet within the confines of a capillary. So I think it illustrates quite dramatically the thinness of this epithelial wall. Once again, this is the junction between two type 1 pneumocytes. This particular section is a transmission electron micrograph showing the following features. This area here, and it's being crossed by the arrow, and now pointed to by the arrow, is the cytoplasm of a type 1 pneumocyte. The cytoplasm of a type 1 pneumocyte lining the alveolar interior is also shown here, and also shows the presence of one of these little bit of myelin body or surfactant regions. This region here is the junction, a junctional complex between the type 1 pneumocyte shown here and the type 1 pneumocyte shown here. So this is the junctional complex and forms those ridges as seen with the scanning electron microscope. This region here is an endothelial cell nucleus. It has a junction here with another portion of an endothelial cell that extends from here to here, and then another endothelial cell, of course, goes off in this particular direction. Or I guess it's going up here and it's sort of overlying. We sort of have a little bit of overlapping of these particular endothelial cells. Once again, this is the blood-air barrier area, endothelium, type 1 pneumocyte, the epithelium, and if one looks carefully here, one can see the fused basal laminae, or the fused basement membranes, representing each. There's a little membrane coming of the type 1 cell and of the endothelial cell, and there's a slight little dense line, the interphase, where these two membranes fuse. So this gives you an idea, at least with the transmission electron micrograph, of what these blood-air barriers look like. Another one is shown here. This particular figure represents a transmission electron micrograph through a type 2 pneumocyte. This is the type 2 pneumocyte here, this cuboidal cell, and if you can envision what was observed earlier in a section of light microscopic material, this is that cube-like cell. It has a junction here with a type 1 cell, the cytoplasm of which is illustrated by the arrow going this way. It also shares a junction here with another type 1 cell going in the direction as illustrated by the arrow. The lumen of a capillary is shown and is being crossed by the arrow at this particular point. And if one looks at the interface between the endothelial cytoplasm and the epithelial cytoplasm here one can envision two fused basement membranes coming from each cell type. And it courses around that type 2 cell, of course, which lies upon it. That vacuolated material, as seen at the light microscopic level, appears in electron micrographs as these myelin or laminated bodies. These types of bodies are those bodies that contain surfactant, which is being produced by the type 2 pneumocyte. So all of these dark laminated structures, hence they're called laminated bodies, or myelin bodies, characterize this type 2 pneumocyte, because this is the position that surfactant is thought to reside within these particular bodies within the type 2 pneumocyte. So this is a classic example of a type 2 pneumocyte, a surfactant producing cell from the alveolar wall. Finally, this is a section stained with an Alcyon blue dye, I do believe, illustrating the pleura on the external surface of the lung. So this blue type of material, as indicated at the tip of the pointer, is actually some fine collagen fibers that have been stained. to illustrate this delicate connective tissue making out the exterior border of the lung. So this is collagen stained blue, and then you can see overlying it and covering its external surface is a simple squamous lining epithelium. This is the mesothelium on the exterior surface of the lung. So this would be equivalent to the visceral pleura of the lung, that simple squamous lining epithelium of mesothelium and a bit of underlying connective tissue, which in this case is stained blue. Obviously also contained within this connective tissue are going to be elastic fibers, which aren't demonstrated, as well as a delicate network of reticular fibers. So this is the pleura of the lung, the visceral pleura lining or covering its external surface.